بسم الله الرحمن الرحيم. النهارده هنتكلم على ال second part of protein classification introduction to functional protein. المرة اللي فاتت كانت introduction to EMBL. This time to an introduction to functional proteomics. Protein domains. What are protein domains? Domains are distinct functional and or structural unit in the protein. قطعة معينة في البروتين، مثلا الأبو إنزيم، الكو إنزيم، بس الكو إنزيم لا، الأبو إنزيم. الأبو إنزيم is a protein، أكيد mainly protein. ممكن يبقى له فانكشن معينة، وممكن يبقى الستراكشر بتاعه بطريقة معينة عشان يقدر يقوم بنفس الوظيفة بتاعت هذا الإنزيم. Usually they are responsible for a particular function or interaction contributing to the overall rule of protein. Domains may exist in a variety of biological contexts where similar domains can be found in proteins with different functions. يعني الدومين دي ممكن تبقى موجودة في أكتر من نوع من البروتين، بس كل واحد منهم له وظيفة مختلفة. Domain example: protein can composed of multiple domains. Often the individual domains have specific functions such as binding a particular molecule or catalyzing a given reaction, and together these contribute to the overall rule of the protein. Sequence features برضو ده another way of protein classification. What are sequence features? Sequence features are groups of amino acids that confer certain characteristics upon a protein and may be important for its overall function. Such further include active site زي الانزيم له active site, binding site, post-translational modification such as in methyl group, benzene ring, whatever of additional part for the protein. Repeats probably a given part of the protein and repeats several times, 2, 3, 4 times, and so. What are sequence features? Active site, which contain amino acid involved in a catalytic activity. For example, the enzyme lipase, which catalyzes the formation and hydrolysis of fats, has 2 amino acid residue, histidine followed by glycine, that are essential for the catalytic activity or catalytic activity. Binding site contain amino acids that are directly involved in binding molecule or iron, and so, as an example, like iron binding site in hemoglobin. Post-translational modification. This is a third way for feature sequencing, which contain residue known to be chemically modified, phosphorylated, acetylated extra, after the process of protein translation. Sequence feature various domains. Sequence features differ from domain in that they are usually quite small, often only a few amino acids long. Whereas domains represents entire structure or functional unit of the protein. Sequence features are often nested within domains. Proteins can also be classified according to the sequence feature they contains. For example, ferredoxin, which sulfur iron protein, a carrier for iron, and it has a sulfur part in their sequence, such as cysteine and cysteine, and so. That mediated electron transfer in a variety of biological redox reaction, including the photosynthetic processes. They can be divided into several groups according to the nature of their iron-sulfur clusters. What are sequence features? In the 2Fe2S ferredoxin, which bind a cluster of 2 iron and 2 sulfur atoms, there are 4 cysteine resistant residues. Cysteine is amino acid has sulfur atom, and cysteine has 2 sulfur atoms, 1 bond and 2 bonds, involved in iron-sulfur binding. The next emerge resistant, the 3-dimensional structure or tertiary structure of a plant type ferredoxin with its 2Fe and 2S cluster. The conserved cysteine residue that help from the binding site are highlighted in red, such this part, this part. اللي متعلم عليها باللون الأحمر أهي، دي ودي ودي ودي، ولو فردنا أكتر هنلاقي تاني. The iron and sulfur atom bound to the cysteine are displayed as spheres. Quite enough this lecture, and we'll meet for another lecture inshallah. Thank you for listening.